Hello and welcome to the next in the JCI series, Conversations with Giants in Medicine. I'm Ushma Neal and I'm delighted to be joined today by Aaron Ciechanover of the Technion Israel Institute of Technology in Haifa, Israel. For decades until Ciechanover and his colleagues appeared, the majority of scientific attention was paid to the synthesis of protein, working out just how DNA produces proteins with its associated RNA machinery. Little research was dedicated to how proteins were degraded and removed from the cell. Enter onto the scene a young Aaron Ciechanover to uncover and describe the elegant ubiquitin proteolytic system. For his description of the ubiquitin system, which we hope to hear a little bit more about today, Ciechanover shared in the 2004 Nobel Prize in Chemistry. I'm looking forward to hearing more about the man behind the discovery. Thank you for joining us so much today, Aaron. Can you start by perhaps telling me a little bit about where you grew up and the influence of your parents and how you discovered science?
So I was born in Israel in 1947. It's the year that Israel was declared as an independent state, directly into the War of Independence. And to parents that immigrated from Poland as kids. So they got to know one another. They met in Israel. And the families left Poland because of rising anti-Semitism. Their parents felt that this is not the place for them, and they immigrated to then, not Israel, but British Palestine. And I grew up in a home, I would say, blue collar, very medium, low salary. My mother was a clerk, my father was a clerk, then he studied law. I have had and have an older brother, 14 years older than I am, that at the end I was very lucky to have this age difference, because when my parents died, he kind of adopted me. And I grew up in a small city on the slopes of the Carmel Mountain. The mountain and the trees and the nature were just behind home, so maybe this is part of my falling in love with the nature early on. And the one thing that I truly remember at home, that we didn't lack food, but it was really a very modest home, but flooded with books. I mean, we had libraries, wall to wall, floor to ceiling, libraries, Jewish books, law books, less science, because I was kind of the only one in the family that was interested in science. My parents taught me to read and write early on, and they made sure that I will read and be curious. And so I remember all the time kind of a scholarly. My father spoke 7 fluent languages. And though he was a lawyer, he was mastering algebra and geometry. He helped me doing my homework. And it was kind of a scholarly environment, but very free. Yeah. My brother is a lawyer, my father is a lawyer, and so I grew up to become the black sheep of the family. But it was really books, reading, knowledge, curiosity.
So what led you towards an early interest in science, or if there was an early interest in science?
There was a very early interest in science. Again, maybe you cannot pinpoint it, but maybe it was nature. You know, I was walking on the mountain, behind our home, drying flowers and plants. Actually, there was a major incident that I even mentioned in my biography. My brother had a big Jewish Talmud, which is the old scholarly codex of laws that accumulated along hundreds of years when the Jews were in Babylon, in the diaspora. And these are very, very heavy books. So they were ideal to dry plants. And when my brother discovered it in one of his visits home, because he was older than I am, he got an apoplexia. I mean, he thought that the next thing that he's going is to murder alive his young, crazy brother. But, you know, lizards, skeletons. So it was more taxonomic type of biology, descriptive, flowers, plants, animals, skeletons, nothing mechanistic, but also biology at that time was not mechanistic. So I think that, and then when my brother went for the first time abroad, when I was like 11 years old, I asked him to bring me a gift, microscope, I still have it. My first microscope, tiny, small microscope, and I started to do my own experiments. Peel onion, and you know, putting the small the layers under the microscope to see the cells, pouring on it water and blowing it of osmosis, then pouring salted water and and pouring and shrinking it and so on and so forth. And then I remember punching myself with a needle and smearing blood on a cover glass. And so I was always there. And when time came to decide in high school, I majored in biology.
Okay, so what led from biology to medical school?
Well, this was a mistake that I then corrected. I somehow felt very interested in diseases and in the human body and the complexity of the human body. And there was also an opportunity by the military, because in Israel you have to serve a national service, to first study and then go and serve in your profession. And the army was obviously less interested in biology than in medicine, in its physicians. So I think that my curiosity about the human body diseases brought me to the medical school of the Hebrew University in Jerusalem. And the first years were very exciting because we studied biochemistry, histology, anatomy, the real secrets of the human body. But then when we approached the clinic, I started to feel restless, that it's not really me there in the clinic. I won't say that there was a pressure at home, but I think that my parents talked to me a lot about medicine and being a doctor. Somehow it's important. Medicine and Judaism, not Judaism, and being a Jewish somehow go together, being a doctor in the family. I don't know why, but. So I went to study medicine, but then I felt restless and I decided to take a year off to do biochemistry, and I immediately fell in love. I mean, I knew that that's it. But then I had to complete my medical studies, and I went to the military, served as a military physician, served in the war during battles, and so on. So I did everything that I had to do. But then, after being discharged and a short stint in surgery, I was still kind of deliberating what I really want to be when I grow up. I landed up safely in biochemistry.
So it was after medical school and your stint as a surgeon that you first came into Avram Hirsch?
Yeah, but I did a year during medical studies, and during my internship I already did my medical thesis. Medical students have to submit a small research thesis in order to graduate. So this I did with Avram Hershko, who just returned as a young assistant professor, non-tenured young assistant professor, and I heard a lot about him before. And then we kept our ties when I was in the military along the way, and when I completed my military combat physician service, I decided to do my PhD with him, and it was the right time, the right person, the right whatever, and luck, and we discovered the ubiquitin system.
Okay. How did you come across that idea to start studying protein degradation?
Actually, it was Avram's. Avram was also thinking about that everybody is doing protein synthesis, mesmerized by the genetic code. And he started already with Gordon Tompkins to work on protein degradation, actually corroborated earlier very paradoxical data, or thermodynamically paradoxical data, that degradation of proteins require metabolic energy, which didn't make any sense. When we eat protein, when we are going to a good New York restaurant, we are eating proteins in order to derive energy for them. So it didn't make any thermodynamic sense to invest further energy in order to degrade a high energy macromolecule like a protein to amino acids. But this was discovered in the 50s already. And Avram further corroborated it. And then there were signs in the literature, very important signs or milestones, that it's not the lysosome that degrades in a programmed, timed manner intracellular proteins, and we honed on those signs and on those milestones, collected them, glued them together. They made a very strong suggestion that the lysosome is not the entire story, and Avram decided to go into it, and he offered me that. And I thought that this was a very nice, adventurous offer, because I wasn't still sure that I really want to do science. You have a profession. People even told me that I'm good. I cannot judge for myself in surgery, and I can handle the scalpel very nicely. And I wasn't sure, but what was nice about Avram, I thought, that I was interviewed by other potential mentors, and each of them was very sure about what we should do, purify this protein, do that, do that. And for Avram, he said, no, there are milestones, but I don't know what I'm going to do. We have to discover something, it's all new. And I thought that this is adventurous, and I am an adventurer by nature. And I thought, wow, let's take it, a year or two. If nothing can happen, if nothing will happen exciting, you can always go and operate and go back to the surgeon, to become a surgeon, to the operating theater. And then after a year, Avram was on sabbatical. I was staying behind in Israel. He came to Philadelphia to work with Ernie Rose, who became our third collaborator. And things started to work out, and we had exciting findings, not realizing at all how important they are. And then I made a final decision to stay.
How did you hit on using cell-free extracts of red cells?
It's biochemistry. The red cell is another story. So we had the science that it's not the lysosome. If we are mentioning the lysosome, you know, the discoverer of the lysosome just died a few days ago. Christian Dediv was a very good friend. The lysosome cannot provide the specificity needed for protein degradation. You know, some proteins have to be degraded at a certain time point. Later on, the wonderful discovery of Tim Hunt and Lee Hartwell and Paul Neres on the cell cycle, that, you know, proteins are stable all along the cell cycle, and bang, they are degraded within a short window of time. So the lysosome couldn't have explained it. There were holes in the lysosomal theory of Christian Dediv. And so we are biochemists, and we decided to go after it. So the one reason to choose the reticulocyte is that when you are doing nucleated cells, the lysosomes spill over the proteases, and the background is huge, and the noise over the signal is like almost covering everything. You have a little blip. And the reticulocyte is a great cell, and because it doesn't have a lysosome anymore. During its maturation in the bone marrow, it expels the lysosome, it throws them out. But it nevertheless involved in extensive protein degradation of all its machineries, leaving behind only hemoglobin, glycolysis, glutathione, BEN3, holding the membrane, maintaining the membrane. And so we picked it up, again, based on milestone. There were other scientists that knew that the reticulocyte is like that. And Fred Goldberg at Harvard Medical School even preceded us discovering the cell-free proteolytic system in reticulocyte. Well, we were working on it already, but then our roads diverged. We took a completely different direction. And as a biochemist, you know, what are you doing as a biochemist at the old times when there is no cloning? You go to the cold room, you go to the columns, you go to chromatography, and you purify the elusive system. And, you know, we purified and purified and purified. Biochemistry at its best.
You mentioned Ernie Rose, and
Yeah.
that Avram was doing a sabbatical with him, and
Right.
you were still in Israel.
Yeah.
And it was at this point that a colleague of yours suggested heating the fractions, correct?
Right.
As you were chasing for what are these components in the split fractions in your
Exactly.
biochemistry?
So Avram was on sabbatical, and I was alone in Haifa, and I had another mentor, a substitute mentor, Michael Fry. And the problem is in the reticulocyte that hemoglobin is like 85-90% of all these proteins, and you cannot get rid of it. Though it has a discrete molecular weight, you know, it's spread all over the column, it reddens it, it's like bloodshed. I mean, so we wanted to get rid of it. We knew that the activity is there because we had an assay, but we could never get rid of hemoglobin. So the idea to heat it was that it's not a protein, probably. So hemoglobin will precipitate, and if it's a sugar or something, it may stay afloat. And we heated it. We didn't heat it, actually. We boiled it. You know, it bubbled in the lab. I mean, like you take a pot and you bubble it, and then you take a filter paper, the mud precipitates, you know, and the clear fluid, the clear yellowish fluid, had all the activity we wanted. It's unbelievable. And then we couldn't believe it, so we had to show that it's a protein. So obviously it was sensitive to proteases, it was high molecular weight, didn't get out of a dialysis tubing. All this, I would say, awfully embarrassing, simple experiment that led us to the discovery. Really simple experiments. Unbelievable.
So this was one of your aha moments, but
This was one of the few aha moments.
you've mentioned that when Avram was off on a sabbatical with Ernie Rose, then you joined him at different times. And he provided some of the other aha moments, given that he was coming at the problem from a different direction.
Yeah, it was really a huge combination of excellent protein chemists that Ernie Rose, an enzymologist. What we discovered, so we heated it up, we purified it. We published the first paper in BBRC, Biochemical Biophysical Research Communication. Actually, it was totally rejected from the JBC, from the Journal of Biological Chemistry. was, I mean, torn not into pieces, but into dust. They didn't leave one word in place. And then I remember Avram called Bernard Horeker, who was the editor of BBRC, and he was interested. So we sent it to BBRC, the first paper. And then we purified this active heat-stable ingredient, heat-stable polypeptide. And what we found already in Haifa, that upon incubation with a crude extract, once we iodinated it, we labeled it, it's conjugated to other proteins to generate high molecular weight adducts. And then we didn't know what it is. This was too difficult. It was a post-translational modification of a protein attached to other proteins. It wasn't clear that it's related at all to what we are looking. And then we went to, in our visits, it was the summer of '79, in another visit to the Fox Chase Cancer Center, to Ernie Rose, and Ernie helped us to find that it's a very stable bond, because initially we thought, wow, it's a, maybe a gentle association, some complex that the protein is added to, and we wrapped it up with cotton wool, biochemical cotton wool, glycerol, DTT, all kinds, that it will not disintegrate. But then Ernie convinced us, I remember the conversations with him were fantastic, that it's a peptide bond. It's a very stable bond. He said, Aaron, do your reaction and put on it hydrochloric acid, or put on it sodium hydroxide and see whether it's stable. And wow, I put it back on the column, completely stable. And and then people there in the Fox Chase Cancer Center, Martin Niemer, Leonard Cohen, worked on histone modification. And they said, they talked to Avram, to Ernie, to me, they said, there is a protein which has the similar molecular weight to yours. We called our protein APF1. We didn't know it's ubiquitin. ATP-dependent proteolysis factor 1, because meanwhile, we started to separate the system to more and more factors. So we had APF2 and APF3 and 4 and 5 and 6. So we established the whole nomenclature in the lab. And they said, APF1 may be ubiquitin. And because ubiquitin is a small protein that is conjugated only once to histone, it doesn't promote its degradation, but maybe you'll try. We have some purified ubiquitin, and you will see whether your protein is ubiquitin. And we did amino acid sequencing, and it turned out to be indeed ubiquitin. And then Arthur Haas and Mike Urban and Keith Wilkinson did wonderful work. We published it then back to back. And they did find indeed that purified ubiquitin came from the work of histones that came from completely different source. Is identical in its function to the protein that we discovered. So the two worlds converge. But I think that the most important thing here was that the bond, the linkage between ubiquitin and histone is a peptide linkage. It's an isopeptide linkage because it's angular, but it doesn't matter. Chemically, and in order to generate a peptide bond, you need energy. So here we solved another mystery, why energy? And then we discovered the activation mechanism. Actually, we went to the Rockefeller Institute. We consulted with Fritz Lipman. We followed his work on bacterial short peptides that are made not on ribosomes, but rather on big complex, bactericidal peptides, gramicidin S, for example. And we followed his technique step by step, and we found, deciphered at the molecular level the activation mechanism. So it's people, knowledge, time, luck, place, whatever you like. I tried to reconstitute it, and it was really majestic. How can so many things fall at the right time, at the right place all together, and bang, the puzzle is solved in like a blow.
Well, given your excitement, even just from the way that you're describing it, what then motivated you to go do your postdoctoral science with Harvey Lodish at MIT on the transferrin receptor? You sort of put ubiquitin on
by chance. It also became a classic. I decided to leave Avram because I felt that I need to be independent. If I stay, Avram actually didn't want me to leave. He said, I'll give you a position, everything, you'll be independent. But I needed to prove to myself that I can do it completely away, because then I realized that all my life, I will be accompanied by his shadow. Doesn't matter that I'll be independent. The people say he grew up with him, he never left, he never did. And I was awfully concerned about it. I really wanted. And also I wanted to be an independent in a different environment, exposed to a different atmosphere of thinking, culture, people, but I decided not to leave ubiquitin and proteolysis. So I wrote to two people only. I wrote to Ginter Blobel, who worked on signal peptide and the cleavage of signal peptide. At that time, I think Peter Walter was at the lab. And Ginter, then we had another funny conversation that he told me that he was so sorry he didn't take me. He said, no, I don't have spot, I don't have space, I don't have money for the next 3 years, whatever. So, and Harvey worked on viral proteins, on polyproteins, on polyprotein that cleave themselves. So the polyprotein translates a protease that then kind of being bent and cleaves the mature proteins out of the polyprotein. And I thought I want to stay in proteolysis, but something else, and then work on also on the ubiquitin system. I had some findings that I had in the lab that were only mine, and I said to Avram, I'm going to take them with me. I'm not going to leave them behind, so if I have time during my postdoc. And then I came to Harvey and I started to talk to him. At the time he didn't work anymore on the polio, though I wrote my proposal, my fellowship on the polio. And he said, why won't you do something completely else? And I don't regret it. We started to work with Ellen Schwartz, who was also a fellow at Harvey's lab at the time, on receptor-mediated endocytosis. And we discovered the whole story of delivery of iron into cells and the recycling of the apotransferrin. Fantastic. I worked with Alice Dautry-Varza. She's now the director of the Pasteur Institute together, and which became a classic. But more than it became a classic, I really was exposed to cell biology, to modern cell biology, culture cells, microscopy, receptor-mediated endocytosis. It was the golden era of cell biology. Not that it's not now, but the discovery of intracellular routing, addressing of vesicles, and it was fantastic. It was really a huge experience, and I still speak the language, and I don't regret it for a moment, but then gradually.
Well, gradually.
Slide it back.
Yeah. Meaning that you also, during that time, were interacting with Alexander Varshavsky,
exactly.
who drew you back into ubiquitin.
Yeah, Alex was interested. Alex was at MIT, and he approached me. He knew that I'm there, and Dan Finley. And Dan was actually an undergraduate in his lab, and then he started his graduate studies. He was just starting his graduate studies. He's now a professor at Harvard. And Alex worked on it, and Alex discovered cell cycle arrest mutant in which the ubiquitinated histones goes away. So he didn't discover the going away of the ubiquitinated histone. This was discovered by the Japanese researchers who discovered the cells. But then he said, what can it be there? And we kind of discussed it together, and we said either they lose their ability to ubiquitinate or they accelerate their ability to deubiquitinate. And it made more sense that they lose their ability to ubiquitinate, because mutation typically is a loss of function rather than a gain of function. And we made extra classical biochemistry, and we discovered a mutation in E1, the first enzyme, activating enzyme in the ubiquitin system. We were also lucky, because if it would have been a mutation in E3, for example, it would have taken another 30 years to discover which E3 is there. But E1, there is only one. So we were really lucky. Luck. I don't know. What's
luck
about
favors
luck?
The prepared
We'll talk about
mind.
luck.
Yeah.
Luck is something else, very complicated. Lucky are the lucky people. So we discovered the mutation and the thermosensitive, but the most important thing there was, or not the most important, one of the important things there was that these were cell cycle arrested mutant. So then we connected. We didn't know mechanistically what happened, because nothing was known about the cell cycle at the time. But we connected the ubiquitin system to cell cycle, which turned out later to be correct, both from the work initially of Mark Kirshner and then Avram Hershko. And now we know that the ubiquitin system is driving basically the entire cell cycle by programmed and timed regulation of many regulators, degradation of many regulators. But this was the first indication. So it was a prediction. We made a prediction in the discussion. It was not a finding, let's make it clear. And we published 2 papers, but meanwhile I did also some other work on, I pulled from the drawer my findings from Israel about the requirement for tRNA in the proteolytic process. We thought that it's linked to protein synthesis, but then it turned out that it's important for marking protein for the degradation. by adding to them an extra amino acid. And basically, I became a freelancing postdoc at Harvest Lab. He didn't know anything about ubiquitin, but he was very helpful. I mean, he wasn't, he said, you're on your own, do it. He helped me a lot, and I owe a lot to the independence that Harvey gave me during my postdoc fellowship, and I was ready to go.
And
many deliberations to stay in the United States, but ended up in...
You did decide to go back to the same institute as
To the same institute, but completely independent. Now I felt confident. Nobody can say, well, I didn't do it on my own. I had my publications with Alex. I had my own publication. I had publications on receptor-mediated endocytosis. So I felt, and I brought my own project with me. And I went back to the same department, but I developed my own career, and Avram allowed it. Obviously, I was an independent assistant professor, and that's it. The rest is history.
Does ubiquitin still motivate you 35 years after you first?
More than ever.
Yeah.
We are not there. We are not even at the beginning of it. Like anything else, we are not there yet. The ubiquitin, it's a system. Remember, it's not a protein. Even proteins are a system. Think about P53. Nobody knows anything about P53. Very little we know. Every day something, one protein. The ubiquitin system is almost 2,000 components, 7% of the human genome. It's a post-translational modification. The beauty of the ubiquitin system that we didn't aim to discover it. We were driven by a biological question, how proteins are degraded. We bumped into it. So I think that the beauty of it is the discovery. But then it turns out that ubiquitin is like phosphorylation, much more complicated chemically, I would admit, because it's a protein, it's not a small molecule, and it serves proteolysis among many other functions which are non-proteolytic, sending proteins to their subcellular destination, activating transcription factors or shutting down transcription factor, activating signaling pathway, NF-kappa B, shutting them down, and so on and so forth. So it's a post-translational modification. It's like a glue, a recognition element in trans for binding of downstream effectors. But the language of ubiquitin is awfully complicated, and we don't even started to read it yet, or to understand it yet. There is monoubiquitin and polyubiquitin. Ubiquitin has 7 lysines. Each of the 7 lysines can be ubiquitinated, short chains, long chains. Ubiquitin-like proteins were discovered, SUMO, NED, mixed chains. You start with the SUMO, you prime with SUMO, you go to ubiquitin. The language is huge, huge language that we have not even started to decode yet. It's huge. It's like DNA. Knowledge is endless. Science is endless. You think about Watson and Crick. The discovery of the DNA, the double helix. It's ended up a period. No, it exploded in our face, and still, mutations, human genome, gene replacement. Think about DNA. I don't compare. You cannot compare pears and apples, but it's the beginning. It's the very beginning. Drug industry. There are drugs already on the market. Patients, more diseases, inflammation, malignancies, neurodegeneration, quality control. Many diseases now are defined now as quality control diseases, and the ubiquitin system is the prime system to clear defective, faulty, denatured, misfolded protein. I mean, we are really scratching the outer layer.
I've seen many of your previous interviews that you've given or things that you've written about how being Jewish has influenced your view on the world, and particularly, can you believe in God and believe in Darwin at the same time?
No, you cannot. You cannot. And it's a very interesting issue because people are asking me, especially, I'm coming to talk to Jews, and Jews are somehow glued by religion. Some are not religious, some are atheists, some are conservative, some are Reform, so there are grading in Judaism. For me, Judaism is a culture. I mean, it's a history of people, it's a language, it's music, it's history, it's the desire for a homeland, and it's morality, and with all the dents that have been poked into this issue and because of politics and recent politics, and so on and so forth. And but kids are asking me, do you believe in God or don't you? And it's tough. It's tough not because, for me it's tough. It's tough to tell them the right answer because they're kids. They don't know about mutations, randomness, survival of the fitting, Darwin, Wallace, not only Darwin. And they are growing up at homes. They are 10 years old, and they are growing to relate to parents that. So you cannot tell them, forget everything you have learned. Tell your parents they are fools or whatever. You have to really approach them that everybody has his own God, his own beliefs. God is about morality, or God is about behavior. God is about peace. God is about human relationship. The prayers are, for me, poets. I'm going to the synagogue just to read to be in the right atmosphere. This Saturday here in New York, I'm going to the service just to be embedded in the atmosphere. It has nothing to do with believing in God or not, but I think that God and Darwin are absolutely in a non-talking terms, and I've sided with one.
Well, speaking of God, one of the other entertaining or intriguing things that I noted on your CV was that in 2008, Pope Benedict XVI inducted you into the Pontifical Academy of Sciences. What was that like, and what is that group?
It's an interesting academy. If you think about religious, only the Pope has a scientific academy, which is the oldest academy on earth. It's 450 years old, which is very interesting. It's dealing with science. We have annual and biannual meeting every 2 years. Very interesting. Actually, the current president is currently a Nobel Prize laureate in physiology or medicine, I think chemistry. I don't remember. Werner Arber, who discovered restriction enzymes. Wow, what a discovery. And he's the president. So you see, it's not, I'm not alone there on that. And there are many Nobel laureates. Actually, the one who really proposed me was a very close friend of mine who just died. Rita Levi-Montalcini, a hero. She got the Nobel Prize in '86 along with Stanley Cohen on discovery of growth factors, how the cell talks to the extracellular environment. She discovered NGF, he discovered EGF. Fantastic, majestic discovery. And she was Italian. And so there are many Nobel laureates there, and the discussions are scientific, on the border of between morality and medicine at time. So I proposed one now about personalized medicine. I'm going to run now one, to chair and to invite people, mostly about bioethical issues in personalized medicine. Sequencing your DNA, what does it mean to know the sequence of my DNA to me, to the environment, prediction, what shall I tell my spouse about my future disease? I mean, obviously, if you want to legitimize abortions as a subject to discuss with, forget about it. So you are walking a little bit on a sensitive, not a sensitive path. You know where you are. You don't want to become a member, it's your choice. But I think that it's important that there will be intertalk between religious people, and we shall need them at the end, because especially in personalized medicine, where so many bioethical issues are behind the corner, huge amount. And so many believers walk on the face of earth, not Catholic, in general, Muslims, Christians, Jews, Buddhists, whatever, there must be an intertalk between scientists, physicians, researchers, and lay people, I would say, and religions, and people that are part of the churches, the different churches, will be part of this dialogue. They must be. Otherwise, we, as scientists, will lose the battle. We shall lose the battle. And I find it fascinating. On top of it, being in the Vatican, I don't know if you visited the Vatican, we are living in the Vatican, inside, so we can see what nobody can see. And that's an important piece of history and of beauty. So you see, another one.
You've spoken in the past about how you did your science because you were driven by scientific curiosity, not by winning prizes or getting honors. But surely sharing in the Nobel Prize with Ernie Rose and Avram Hershko in 2004 in chemistry must have been very sweet. Can you tell us a little bit about that time?
You know, obviously we are happy about it. I think that all three of us, and we did not appeal the decision. But I got it relatively young, and you celebrate for one day, And then you celebrate the second day, and the third day you have to decide what you are going to do with yourself. You cannot celebrate forever. And I decided to do two things. First of all, to continue my science. And we do continue science. Actually, we are doing now, I wouldn't say better or more original science. We are doing I think that we are doing good science now. New paradigms in the ubiquitin field, monoubiquitination as a signal for degradation, collaborations with synthetic organic chemists. We are now being able to conjugate, to synthesize ubiquitin conjugates that we never able before. Milligrams amount of a defined conjugate, a protein, with ubiquitin sitting wherever we want to put it, or even a chain of ubiquitin. It's an extremely powerful tool to understand the proteasome and so on and so forth. So we are doing, I hope, good science. So this was my major decision, because otherwise I will feel that, I was cut off my umbilical cord and I became kind of a spaceship. But then I decided also to leverage my new position to two major issues. Handling, if I can. One is education of kids in Israel and worldwide, mostly by showing them that it's possible, that it's not in heaven, that it's not a Nobel Prize, it's the achievement behind the Nobel Prize, it's their hands reach. There are a zillion of scientists who are better than I am. I mean, they discovered things. It's not the Nobel Prize, it's that you can make a difference, you can make an important discovery. And human beings are doing it with genes, with, that talk to you the same language, that are excited, that can curse in your language, that are familiar with your culture, that talk your language, especially in Israel, that I can talk the language and curse and know the history, and I can share with them values. So this is one thing, and I think that it really excites them. It really excites them. You talk to them at their eye height, which is very important. They come and grab you and take photos with you, and you sign them and you joke with them, and they see, wow, it is possible. And many kids are writing to me back. You made it to me. You made my day, you made my decision, you helped me, you assisted me, you excited me, you are illuminated my road. So, and it's important. And I do it on a personal level. I don't write big educational program. I try to talk to politicians, but this is useless. I know that talking to politicians is important because then you can magnify your voice. But I don't know. Everybody does what he likes to do. But talking to the kids, it's fantastic. So doing my science, education, and getting familiar with the Jewish, the opening of the gates of the world. That's here I'm abusing my celebrity, quote unquote, position because I'm invited. I insist wherever I go, especially, and I built very close relations with remote Jewish community. Greece, which is only an hour and a half flight from Israel. I have terrific relationship with the Jewish community there. I speak in sermons in their synagogue. I'm visiting them. There were 500,000 Jews in Greece. The Salonika was a Jewish city. All the harbor was operated by Jews. They were all perished. 3,000 Jews were left out of 500,000 in Greece. How they can lead Jewish life? How can they marry? How can they have kosher food? How can they do anything? It's really fascinating for me. And I think that I feel when I come and talk to them that I blow unbelievable wind in their sails. They inflate it. I inflate their sails. And it's really a very good, I feel belonging. I'm not, many scientists are doing science. I feel deeply rooted in my parents' heritage, in what I am, in where I come from. It's important to me.
One quick last question then. If you were not a scientist, what do you think you would have done with your life?
Well, physically I'm continuing with ubiquitin, but what really, let me put it this way. Let me reword or rephrase your question in a different way that you may not have think of it while asking it. I would like to live my life not in one chunk of 80, 90, 70 years, however God or whoever assigned me, allotted me with, but in pieces. Living 5 years, dying for 5 years, and seeing the progress in science. We have entered a very exciting period now. The human genome understanding what's going on. For now, it looks like a swamp. So, I mean, now I think that we start to have tools to understand real biology, and that's exciting. So, I don't know what I would have done if I would have been, I probably would have gone to a neuroscience. But the ubiquitin system is in the brain, and so we are working on it also in the brain. We are now working on maintenance, the ubiquitin system, and maintenance of synaptic integrity. So we are pushing our hands. You know, I'm driven by curiosity, so whatever is interesting.
Well,
as long as the name ubiquitin is pegging the top of the page, it's in the title.
Well, thank you so much for taking the time to talk to us about your life. I really
you.
appreciate it.